and welcome to the Activating an Immune Response Vaccines section of the BioBasics course. In this section, we'll give an overview of how vaccines work. We'll describe the process of making the annual flu vaccine. We'll discuss a universal flu vaccine that's currently in development, and we'll summarize the different types of vaccines. Finally, we'll explain how a novel type of vaccine, DNA vaccines, work, and give an overview of how a viral cancer vaccine, also referred to as an oncolytic vaccine or an oncolytic virus, works. It's just first a little bit of history. So vaccines were discovered in the 1700s by a British scientist called Edward Jenner. So Jenner had this very important observation that milkmaids did not get smallpox at a time when smallpox was devastating most of the population. So people were either dying from smallpox or they were left with disfiguring scars from the disease. And so Jenner hypothesized that milkmaids did not get smallpox because they somehow were protected by their exposure to a similar virus that cows got, cowpox. So milkmaids were, their job was to milk cows. They were around cows every day. So when cows got sick, they were exposed to that. And so this formed Jenner's hypothesis that if we can expose somebody to the cowpox deliberately, and so that's what he did, he exposed a young boy to the cowpox virus. He let that boy recover and then deliberately infected him with smallpox. And the boy did not get smallpox. And so, by today's standards, this is not the kind of experiment we would do because we don't have all the ethical controls in place. But Jenner's observation was very important, and he was correct that by exposure to a related virus, people would be prevented from getting this deadly smallpox virus. And so since that time, really since about the mid-1950s, we've developed a whole range of different viruses, again, using more sort of standard controlled clinical trials of monitoring for safety, et cetera, treating patients with inactivated pathogens to achieve an immune response. And so this just shows some of the statistics of where we would be without vaccines today. And so this just follows directly from what we talked about in the immunology section. So vaccines work because if you can expose somebody with an inactivated version of a virus, and we'll talk about that a little bit more on the next stage, but this initial exposure to the virus allows that person to generate helper T cells, B cells, antibody response. And so now we have memory B and T cells. So the next time we see that live virus, we have a rapid immune response, so a 24-hour immune response to rapidly defeat that virus. Whereas after the first exposure, we have 6-week lag time, as much as 6 weeks lag time, which causes people to become sick and potentially even die from virus exposure. So there's a couple of different types of vaccines. First generation vaccines, this just means those types of vaccines that were used first were whole pathogen vaccines. And so this essentially means we grow up large quantities of a particular type of virus, kill that virus. We can kill it with exposure to heat, exposure to UV radiation, ultraviolet light. We can add chemicals that will kill the virus. All these different ways of killing a pathogen and then injecting that into the patient so that those antigens, those proteins that activate a T cell and a B cell response are present, uh, but the virus is not able to replicate, not able to make the person sick. Uh, Another type of whole pathogen vaccine is a live attenuated pathogen. And so attenuated just means weakened in some way, but still alive. So a couple of different ways of doing this is, for example, by um, growing the virus in the lab at a slightly different temperature. So 36 degrees Celsius is the temperature of the human body. It's the temperature that these viruses have evolved to sort of replicate at. If we adapt them in the lab to a slightly lower temperature, so 34 degrees Celsius, for example, then when these viruses, when are in the human body, they will not survive for very long. They will not be able to replicate, but they will replicate to very low levels. The advantage of this is you get a stronger immune response, essentially. So especially in terms of T cells. So whole pathogen killed vaccines are very good at activating an antibody response, less good at activating that strong cytotoxic T cell response. So some of these live attenuated pathogens do a better job at that. Second generation vaccines, and I just want to point out here that in our use of first generation, second generation, it doesn't mean that we're no longer using the first generation vaccines. So these types of vaccines are still available. It just means that we've also developed other technology as well. That's not necessarily better. It's just a different approach, and different approaches work better than other approaches for different types of viruses. So it's really trial and error for the companies, for the research labs that are developing these vaccines, trying different approaches, see which one will give the best immune response for this particular pathogen. So second generation vaccines include what we refer to as subunit vaccines. So these can include, particularly in the case of bacteria, we have just toxoid vaccines. So the tetanus vaccine, for example, which most people have taken or should have taken. This is a vaccine you take to prevent you from bacteria that you might get if you step on a rusty nail, for example. So standard vaccination. And what is actually protecting you against is the toxin or a protein that's released by that bacteria. And that toxin is what makes you sick. Um, Protein antigens, this just means particular proteins, uh, typically proteins that are found on the surface of the virus. These are the proteins that we're most likely to have an antibody response to. An example of that type of vaccine is the HPV, human papillomavirus vaccine. Gardasil is made against one of the surface proteins on that virus. Third generation vaccines, still in development, haven't quite yet um, uh, been able to bring those to market, are DNA vaccines. And we'll talk a little bit more about that on the next slide. But again, even though the concept of vaccination is not new, depending on the pathogen, it can be very tricky to develop a vaccine. So especially RNA viruses like HIV, Ebola, hepatitis, Zika. These are all RNA viruses. Malaria is not a virus, but it is a parasite, so that can be challenging as well. The RNA viruses are challenging because they mutate very rapidly. And so the rapid mutation of that RNA genome means that the antigens change very rapidly as well, making it hard to make a sort of a one-time vaccine against them. And so these are areas of still ongoing active research to come up with vaccines against those. Although there has been some good news on the Ebola front lately. There's a vaccine now that looks like maybe protective against Ebola. And so making the annual flu vaccine is sort of ties into this idea of why viruses that have an RNA genome can be tricky. So the flu virus has an RNA genome. That means the genetic material is made up of RNA. It mutates very rapidly, so it changes every year. It changes so much that the flu virus that it infected us last year, even if we got sick from it or got a vaccine against it, that is not going to be likely to offer us protection against flu virus that's going to be in circulation next year, because it changes enough that the antibodies no longer recognize it. So that's why we have to get a new flu shot every year if we want to maintain optimal protection. And so the way that flu shot is made is when it is springtime, so March, April, May, in the northern hemisphere, the Center for Disease Control, the World Health Organization, track the flu virus strains that are causing problems in the southern hemisphere, that are most likely to make people sick in the southern hemisphere, because that is their wintertime, that is their peak flu season. And the prediction is that those are the same strains that are going to be causing us problems in the northern hemisphere next flu season, the following winter. So these agencies identify those strains, they select specific strains, and those are the strains that are going to be used to make the flu vaccine. And the way that vaccine is made is the selected strains are mixed with what are called non-virulent strains. And non-virulent just means that these strains don't make people sick. So not all viruses make people sick. And the idea is, if we mix the non-virulent strains with the virulent strains, so we're going to inject both of these strains into the same egg, and yes, that is just a regular chicken egg, those strains are going to recombine. So we're going to incubate them. They're going to literally swap genetic information with each other, resulting in new gene combinations. And so this is essentially a way of attenuating or weakening those selected strains. So the same antigens will be present, just in a different combination, which is likely to be less virulent, less likely to make somebody really sick. So we're going to take these recombined strains, and then as an additional safety measure, we will further inactivate them. So again, we'll subject them to heat treatment, to UV light treatment, any of these different methods to make sure that this attenuated virus is inactivated and is not going to make us sick. And then purification, testing, FDA approval. And of course, so for the flu vaccine, essentially it's the method of production that's approved. Because there's such a rapid turnaround, we don't have to do additional testing each year. If a company is making the same type of vaccine they've made the year before using the same methods, it already has FDA approval. But of course, it does still have to be purified. Like any type of biomanufacturing, it does need to be purified and tested before being shipped to pharmacies and doctors' offices. And so this is just an overview of making the annual flu vaccine. This is also kind of helps to explain why the flu vaccine is not always 100% effective. And that is, again, because it depends on selecting the right strains. We are not always able to predict with 100% accuracy what strains are going to be the correct ones to include in that vaccine. And so our virus may be more or less effective based on that. So we want to develop universal flu vaccines, that is, flu vaccines that we don't have to administer every year. And so different researchers, different academic labs, different biotech companies are looking into this. And so essentially what they're trying to do is, rather than target the head, so the protein that's most often gives us an immune response is this protein called hemagglutinin, which is on the surface of the virus. And typically, an inactivated flu virus vaccine is going to cause the recipient to generate antibodies against this head portion of the vaccine. And so these vaccines can be effective, can be very effective if we pick the right strain, but not for more than one season because of the rapid mutation. So in development is this idea of a universal vaccine. So targeting the stalk region, this stem here, that is more conserved, or that mutates less frequently than the head. And so the approach that's being used is identifying specific epitopes, or short segments of protein. So short sequences of amino acids, 5 to 20 amino acids within this stalk region, combining those and using that as a vaccine. And so again, preclinical studies testing in animals suggest that this approach can generate a robust immune response to the virus. And because that region mutates less frequently, it's more likely to last longer. It probably would not be a lifelong immunization, but the goal is for at least 5 years or so. And this is currently being developed by BioBasics and is in phase 3 clinical studies. And another approach is targeting internal antigens. So again, we talked about antigen presentation. This would allow us to generate a T cell response as part of the vaccine against some of these internal antigens that may be presented on an infected cell surface. Okay, so now back to DNA vaccines. I had mentioned this briefly on the slide that talked about the different types of vaccines. And the idea here is, rather than introduce a whole pathogen or one protein from that pathogen, let's introduce one gene from that pathogen, a gene that codes for an antigen, and inject that into patient cells. So here we can isolate a gene virus, say, insert that into a plasmid, which is a vehicle for delivering foreign DNA to cells, and then inject that into a patient's muscle cells, again, similar to where we inject the flu vaccine normally. And then the idea is that patients' muscle cells would take up that viral gene and make the associated viral protein, and then we would mount an immune response against that protein. And so again, these are in clinical development. The holdup is our cells don't necessarily want to take up foreign DNA. Just injecting it with a syringe, the way we would inject a traditional vaccine, doesn't seem to deliver enough of the DNA vaccine to have an effect. So now there are devices that are being tested for delivering the viral DNA that combine traditional needle injection with electroporation. So basically send a pulse of electricity along with that injection, and this induces the muscle cells to take up this DNA and then subsequently make that protein. And so one of the reasons companies are pursuing this approach is it has a couple of advantages. One, again, a better immune response. That means typically a longer antibody response, a better cytotoxic T cell response, because having the patient's cells actually making the viral protein more closely mimics a natural viral infection. So you're going to get that antigen presentation that's necessary to activate T cells fully. A lower cost manufacturer than so manufacturing protein subunits, manufacturing full viruses is very time intensive, very expensive process. Manufacturing these plasmids is less expensive. Um, the plasmids are also much more stable. This means they don't break down as easily. So, you know, we would likely want to refrigerate these types of vaccines where possible, but if we wanted to use these, for example, in developing countries where refrigeration wasn't readily available, and refrigerated trucks for distribution maybe weren't available, these types of vaccines would be able to withstand these different temperatures much better than traditional vaccines. So these are just some of the reasons that we are pursuing DNA vaccines. And this gives the name of some of the vaccines that are being pursued using this technology. And now a really exciting new type of cancer therapy. And so this is referred to sometimes as a viral cancer vaccine. We also sometimes hear the term oncolytic virus, and onco refers to cancer. Lytic means the virus is going to lyse open these cancer cells. And so essentially what this product, and there's several different companies are pursuing this type of product. There's one on the market. And the way it works is it is a modified HSV, so herpes simplex virus. It's modified in a couple different ways. One, the genes that cause this virus to cause cold sores have been removed. It is also expressing GM-CSF, which is one of those cytokines, inflammatory cytokines. This is just a very general, very strong inflammatory cytokine that encourages the development and the production of more white blood cells. So it helps to rev up the cancer patient's immune system. It's also been modified so that it can only replicate in tumor cells. So essentially what happens is we inject it directly into the tumor. It infects and replicates in those cells, and it releases this powerful cytokine. As part of its replication cycle, it replicates so much that it directly kills some of the cancer cells. So lysing basically means burst open. So the virus reaches such a high volume inside of these cells that it bursts open and then goes on to, so it directly kills the cells when it bursts open, and it can then go on to infect other tumor cells. It's also releasing these cytokines, shown here as these yellow dots, and these cytokines help to attract white blood cells to the area that also recognize that there's viruses replicating inside of these tumor cells. I better help get rid of those cells. So it really works by three ways, or by two ways: direct killing of the tumor cells, and then activating a strong white blood cell response against that tumor. And so this particular oncolytic virus has been approved by both the FDA and the EMA to treat melanoma, and it is now in late phase clinical testing for head and neck cancer. It's a very interesting technology. And again, other companies have these types of oncolytic vaccines in development. So this concludes our section, activating an immune response. So vaccines are just deactivated versions of a pathogen that train the immune system, so that when we're exposed to the same pathogen again, we have a very quick response. Because the flu vaccine mutates so rapidly, we produce a new flu vaccine every year. Scientists are trying to develop a universal flu vaccine that would be effective against multiple strains of the virus, so that it would be effective for several flu seasons instead of just one. Different types of vaccines include whole pathogen, pathogen subunits, and DNA vaccines. And again, the DNA vaccines are still in development, but could result in a better immune response and a more stable during shipment and storage. And finally, a very novel type of cancer therapy is the oncolytic or viral cancer vaccine that directly kills tumor cells and also activates a white blood cell response against those same tumor cells. So thank you. That concludes our section on activating an immune response.